What goes in but never comes out? Hi everyone, and welcome back to my channel. If you're new here, I'm Aman Praveen, a second year medical college student in SMMC India. As you can see, the biochemistry of iron can be divided into 8 subcategories. And when this essay or short notes, shows up, make sure you cover all these topics. So the topics we have are the recommended daily allowance, the sources from which we can get iron, and the factors influencing absorption and how the absorption is regulated, and from the absorption side how it is transported and how it is stored in our body and how it's excreted, and then we'll brush up on the clinical aspects also. So the recommended daily allowance is the minimum amount of the substance which we should have in our daily course of meal to have a sustainable life. You see, for a normal adult, it is about 20 milligrams per day, and for children, it's from 20 to 30 milligrams, and for pregnant women, it's 40 milligrams. And then we'll go into the sources from which we can get iron. The sources are leafy vegetables, the liver, meat, jaggery, and cooking using iron utensils. So now we'll be discussing about the factors influencing absorption. As you can see, the factors influencing absorption are the ferrous form. See, iron has various electrochemical forms, the ferrous form and the ferric form. The ferrous form is actually, the state at which it can pass through one cell wall to another. Ascorbic acid. Ascorbic acid is otherwise commonly known as vitamin C, which is found in amla, lemon. It helps in reducing the ferric form into the ferrous form. Thereby, when there is more ferrous form of iron, it facilitates absorption more. Recommended daily allowance for vitamin C is 50 to 75 milligrams per day. You see, these green vegetables has these oxalic acid and phytic acids, which convert this iron which is absorbed in our body, and it converts it into salts which gets deposited, or in turn reduces absorption rate. That is why, even though the Western countries who have lesser iron containing diets have adequate amount of iron in their body, because those inhibiting substances are less in their diet, whereas in our Indian diet we have all these green leafy vegetables and all that, and there is enough iron in our diet. But the problem is we have more inhibitors also. Just like these interfering substances, there are certain minerals which also competitively inhibit. As you might have studied in enzyme kinetics, calcium, copper, lead, phosphates, they competitively inhibit iron. The mucosal block theory. After we have our food, it goes and gets digested, and then it enters the intestine. Okay, so that's the intestine, and ferric ions are passing through it. Ferric ions break down from the food, which is Fe+3, and it breaks down. Now it has to enter into the human body. So how does it enter? You see, these intestinal lumen are actually lined by brush border cells. These brush border cells have these specialized structures called as the divalent metal transporter. What is divalent metal transporter? It basically transports every metal which has +2 charge, which is the ferrous. Ferrous ions have Fe+2 charge, and these detect these ferrous ions, and then it allows it to enter into the intestine mucosal cells. See, these linings have these divalent metal detectors, which join with the ferrous ions, and then thereby it pushes it inside the intestine mucosal cell. Over here, the ferrous ions get again turned into ferric ions. After forming into the ferric ion, it can't just stay as a ferric ion inside the cell. That is harmful. It might lead to neoplasmic conditions. So therefore, it binds with a temporary storage form known as a ferritin, with which it forms, and it's a temporary storage. Whenever there's requirement, this ferritin breaks down and gives it to the body. Other than that, it just stores it just for some extreme conditions. And then from the mucosal cell, it has to enter into the bloodstream to supply various organs which require iron. For that, it crosses the membrane, and after crossing it, and during that crossing also, you need to make sure you remember this one thing: ferric ions cannot pass through one junction. It can't pass through the lumen into the cell or from the cell into the bloodstream. Only ferrous ions can. So therefore, this ferritin ion, which has ferric ion, it breaks down and it again forms ferrous ions, which enter the blood. And after entering into the blood, it again gets back to ferric form, which it binds with the transferrin molecule. You see, the transferrin molecule, we'll study it in transportation. So yeah, this is mucosal block theory. The next is storage of iron. Storage. So what is the main storage form? It is ferritin. Ferritin is the storage form of iron, which is present inside the intestinal mucosal cell, in the spleen, in liver, in bone marrow. This ferritin molecule has 23% of iron, and as you know, ferritin is never, it is never found in plasma under normal conditions, because it's mostly in the mucosal cell or the spleen or the liver or the bone marrow. It increases in the plasma only during iron overload. And then there's another form with which iron is stored, which is the hemosiderin. Hemosiderin is also a kind of a smaller version of ferritin. You see these ferritin molecules, these are proteins which can be deproteinized, denatured. When it gets broken down by the lysozymes, it forms the hemosiderin, which is also a minimal form of storage, which is present in the liver, spleen, and bone marrow. There's a link in the description of my notes. Go check it out. So, regulation of absorption of iron. Uh, we can categorize it into mucosal regulation, stores regulation, erythropoietic regulation, and the ferritin transferrin receptor relationship. The most important is the mucosal regulation. You see, iron present in the mucosal cell can be, stored in it with ferritin molecule, or it can dissociate and enter into the bloodstream. So the transport across the cell membranes, there are two main protein molecules. One is the divalent metal transporter, which transports the iron molecule from the lumen into the mucosal cell. And then from the mucosal cell into the bloodstream, the ferroportin transports it. So these two proteins are the main substances which regulate it, and these are encoded each protein is supposed to be encoded, right? So this encoding of these protein is inhibited by hepcidin, which is produced by liver. This liver, during adequate iron storage, it releases hepcidin, which downregulates or inhibits the ferroportin and divalent metal transporter, thereby causing decreased absorption. In cases of anemia or hypoxia, what happens? We require more RBCs. RBCs require more iron, and for that, we need more absorption. At that time, liver decreases the production and release of hepcidin. And then next is the stores regulation. This is more simple. Whenever whenever the blood iron level falls, what happens? Signals go to the mucosal cell to increase absorption. And then there's the erythropoietic regulation. You see, during anemic conditions, uh, what happens? Our oxygen saturation decreases. That in turn stimulates the kidney to release erythropoietin. Erythropoietin thereby stimulates the bone marrow to increase RBC production, and thereby when there is increase in RBC production, there is more requirement of iron, thereby it increases iron absorption also. And then there is the ferritin transferrin receptor relationship. In this, when the plasma iron level increases, what happens is the gene coding for ferritin increases, because ferritin is the storage form, and since we have a lot in our blood, it has to be removed and it has to be stored. And iron, what happen when the gene is coded for ferritin, more ferritin molecule is produced, which in turn takes back all the iron from the blood and stores it, and thereby decreases the blood iron level. And at the same time, when iron plasma level increases, the gene coding for transferrin receptor stops or decreases, thereby decreasing absorption. Next, we'll be seeing the transports. So, transferrin molecule is synthesized by the liver. It is a glycoprotein and a beta 1 globulin. The normal blood plasma level is 250 milligrams per 100 ml of blood. You see, one transferrin molecule can carry two ferric atoms, and it has 20 polymorphic forms. The half life is 7 to 10 days. And with respect to transferrin, there is another uh, value you need to understand. It is the total iron binding capacity of your blood. It is roughly around 300 to 400 milligram per 100 ml of blood. The other form of transport is ceruloplasmin, which is present in the blood along with transferrin. Iron is a one-way element. It only gets ingested, never gets excreted actually. It is mainly regulated at the absorption level. Very little is excreted actually, and it might be lost during bleeding, like during menstruation or during a trauma. Almost nothing, almost trace amounts only get released in urine, and very little is excreted via the feces. And these iron are the particles which do not get absorbed actually. And this is because the intestinal lining disintegrates and replenishes almost 30 percentage per day. And iron goes and deposits in our skin, and as skin sheds off in a regular basis, and thereby that is a way through which iron gets excreted. Now we are into the final topic, that is the clinical aspects. The clinical, we have two. Main is very low iron, and then there is high amounts of iron. When there is deficiency of iron, it mainly causes iron deficiency anemia. I hope this video was really useful for you. And don't forget to like, share and subscribe to my channel. And I'll be posting much more interesting contents in the near future. So stay tuned and always be awesome.